Hello everyone. So the next topic which we will be discussing is about the biocatalysis. So what do you understand by the term biocatalysis? So biocatalysis is a catalyst. It is a substance which alters to promote the reaction, and a substance, especially an enzyme. So it will be an enzyme. What the enzyme do is it initiates or modifies the rate of the chemical reactions in a living body, and it is termed as a biocatalyst. So what is it termed as? It is termed as a biocatalyst. So they are enzymes or microbes that indicate or accelerate the chemical reactions. So they are that. First, we will discuss about the introduction part, that what is biocatalysis. Then further, we will discuss the functions and the types. So the biocatalysis refers to the use of living systems or their parts to speed up the chemical reactions. In the biocatalysis process, there are natural catalysts such as enzymes which perform chemical transformations on organic compounds. So utilizing the natural or modified enzymes to perform organic synthesis, it is termed as the chemosynthetic uh, chemoenzymatic synthesis, and the reaction which is being performed by the enzyme, it is classified as the chemoenzymatic reactions. So this is about the biocatalysis. Now if I talk about the history, so first of all, this is a diagrammatic representation. So it is a 3D dimensional structure of a enzyme. So biocatalysis, it utilizes these biological macromolecules to catalyze the small molecule transformation. So this is the 3D structure, and it the process of the biocatalysis uses these kinds of biological macromolecules to catalyze the small molecule transformations. So next, about the history of the biocatalysis. So the biocatalysis underpins some of the oldest chemical transformations which is known to humans for the brewing predicates recorded history. So the oldest record of the brewing was about around 6,000 years old, and it refers to the Sumerians. The employment are of centuries. So the most obvious uses have been used in the food and the drink business, where the production of wine, beer, and cheese is dependent on the effects of the microorganisms. So more than 100 years ago, there was biocatalysis, which was employed to do chemical transformations on natural man-made organic compounds, with the last 30 years seeing a substantial increase in the application of the biocatalysis to produce the fine chemicals, especially for the pharmaceutical industry. So since the biocatalysis deals with the enzymes and microorganisms, it is historically classified separately from the homogeneous catalysis and the heterogeneous catalysis. So this, mechanically speaking, biocatalysis is simply a special case of heterogeneous catalysis. So this is about the history of the biocatalysis. The next comes is the factors that affect the catalytic activity of the enzymes. So we will be discussing that in detail. So the first of all, there will be an enzyme concentration which will affect the catalytic activity. So if I talk about the enzyme concentration, then the product concentration increases with the increase in the enzyme concentration. So the graph here of time versus uh, product concentration, it will be around this. So the graph will appear to be like this. So the product concentration, it increases with the increase in the enzyme concentration. The next comes is the substrate concentration. So it has been shown experimentally that if the amount of the enzyme is kept constant and the substrate concentration is gradually increasing, the reaction velocity will increase until it reaches a maximum. So after this point, increases in substrate concentration will not increase the velocity. So this is what is about the substrate concentration. The next is about the effect of inhibitors on enzyme activity. So enzyme inhibitors are substances which alter the catalytic action of the enzyme and consequently slow down or in some cases stop the catalysis process. So there are three common types of enzyme inhibition. There is a competitive inhibition, there is a non-competitive inhibition, and there is a substrate inhibition as well. So these are the three common types of the enzyme inhibition. The next is I will talk about is the temperature. So like in most of the chemical reactions, so what is happening in most of the chemical reactions is the rate of the enzyme catalyzed reaction will increase as the temperature is has been increasing. A 10-degree centigrade rise in temperature will increase the activity of most enzymes by 50 to 100%. Variations in reaction temperature as small as 1 to 2 degrees may introduce changes of 10 to 20% in the results. So this was about the temperature effect. The last is about the effect of pH. So the enzymes, they are affected by changes in the pH. The most favorable pH, the point where the enzyme is most active, that is also called as the optimal pH. So this was about the factors that affect the activity of the various enzymes. The next is about the types of the biocatalysts. So there these types are being shown. So first we will be discuss about the oxidoreductase. So it is the, as the name suggests, so here there will be, they will catalyze the oxidation and the reduction reactions. For example, if I take the example here, alcohol dehydrogenase converts primary alcohols to aldehyde. So this is the example of oxidoreductase. In this reaction, basically, ethanol is converted to acetaldehyde and the cofactor NAD, which is further being converted to NADH. So in other words, we can say that ethanol is oxidized and NADH is reduced. So there is an oxidation and there is a reduction, which in this reaction both the things are coming up. Then comes the transferase. So transferase, as the name suggests, it will be the transfer of functional group. So align aminotransferase shuffles the alpha amino group between alanine and the aspartate. So this is about the transferase. Then hydrolases, it is the catalyze the hydrolysis of various bonds. So what it does, it catalyzes the reaction of the various bonds. So for example here, phosphatases, they break the oxygen phosphorus bond of phosphate esters. This is the example. Lysases, so it is the formation or removal of a double bond with group transfer. So what is it? It is the formation or removal of double bond. So example here will be dehydratase, which remove water in, as in the case of fumarase. So this is the example here. So after this isomerase, so isomerase, it basically what it does, it catalyzes the isomerization changes within a single molecule. For example, triose phosphate isomerase carry out these rearrangements. Then comes the ligase. So it is the removing the elements of water from two functional groups to form a single bond. So this is what is known as the ligases. Then after, the last one is the kinase. So this enzyme in the body attaches a phosphate group to a high energy bond, and it is very important enzyme required for ATP production and activation of the certain enzymes as well. So these are some of the types of the biocatalyst. So I have given the examples of each biocatalyst and what is happening in that particular reaction I have already told you. The next is about what are the disadvantages of the biocatalyst. So there are enzymes that require the narrow operation parameters. So these kinds of enzymes, they display highest catalytic activity. So these are some of the disadvantages of biocatalysts. These enzymes cause the allergies and they are often low specific, they often show the low specific activity. There is an instability which is at extreme temperatures and pH values, and availability for selected reactions only, long development time for new enzymes. So these are some of the disadvantages of biocatalysis. So this was all about the biocatalysis part. These are some of the references. Thank you.